All right, so we're going to get started. My name is Dina. I'm a postdoc at the University of Vermont, and I'm a member of the Women in Cachexia Planning Committee, and we've planned a great seminar for you today. But just before we get started, I have a couple of announcements. Women in Cachexia Initiative has been collecting information about publications, awards, and other announcements that we've been sending out in a quarterly newsletter. So if you have anything great that you'd like to share with the Cancer Cachexia Society population of people that get these emails, please go ahead and click on the link in the chat to share that information with us. We would love to brag about your accomplishments. We also need to advance my slide here. I'd like to just go ahead and take a minute to promote an upcoming trainee talk that we have next week on Wednesday at noon Eastern Time. We have 3 great postdocs. This is organized by the Education Committee. 3 great postdocs will be presenting their research. And then finally, of course, as many of you may know, the registration is now open for the Cancer Cachexia Conference. It's happening in Italy in September of this year. So please go to the website and register for that. Now, finally, it's my pleasure to introduce Dr. Michelle Law today. And it's my pleasure to introduce Dr. Law. After earning her bachelor's degree in nutrition and chemistry from Concordia College in Minnesota, she completed her dietetic internship and earned a master's of science in medical dietetics and a PhD in nutrition from The Ohio State University. During her doctoral studies, she published seminal work describing insulin resistance and cardiac wasting and remodeling in the C26 model of cancer cachexia. She went on to complete postdoctoral research in cardiac physiology and molecular cardiology at the University of Minnesota Medical School. As such, Dr. Law has developed expertise in cancer cachexia, metabolic disease, and cardiomyopathies. The focus of her lab's work is to elucidate the mechanisms of cardiac muscle dysfunction in cancer cachexia and how decreased cardiac performance may contribute to cachexia pathogenesis. Please join me in giving a warm welcome to Dr. Law as she discusses navigating her path in science and in life. And just as a reminder, for questions, please use the question and answer box for any questions you may have for Dr. Law. And thank you. Take it away, Dr. Law.
All right. Well, thank you everyone for having me today, and thank you for that kind introduction. I will share my screen here. All right. Is that visible to people? Yep. Okay. And I will move this over here. And then have this up here. All right, so I'm organizing myself here with all my different screens. All right, well, thank you so much for having me. I'm really excited to be here and have the opportunity to share with you all today in this WCX Stories in Science series. This is a little bit of a different format than I'm used to as far as, being different from a general research talk. I will share a little bit about my research, but I will share my journey, educational journey and training journey, and how I got to the place that I currently am. What my major, what this position is that I'm currently doing, and the research that I do, and how I develop this research agenda, and then some of the lessons that I've learned along the way. So to start, we'll start with the journey, and we'll start with essentially my path from first generation college student to postdoctoral fellow. And I show this, I show this image here because it's kind of a windy road. And I will say that my road has been a bit on the windy side, but I've really found value and meaning in each step of the journey. So I did my undergraduate work at a small liberal arts school up near Fargo, North Dakota, in northwestern Minnesota. And if you'd asked freshman Michelle what she was going to do with her life, I would have said, I want to be a public health dietitian and develop and implement nutrition education programs for underserved communities. And it was really during my freshman year of college that I unexpectedly found that I really enjoyed the science classes. I had always assumed that I wasn't really good at science and science wasn't for me, and I don't really know where that came from, but I really did enjoy the sciences. So I went a little bit more along that track. Although I majored in dietetics, I also took on a secondary major in chemistry. And by the time I was a senior, if you would have asked me, I would have said that I wanted to be a critical care dietitian working in ICU with a nutrition support team. And so I really decided then to go on to become a registered dietitian and did a master's degree and dietetic internship combined program at Ohio State, and it was very clinically focused. But, besides all of the, clinical rotations and things that were part of my internship, I was also able to do thesis research. And so we did a clinical study which looked at a novel, slowly digested carbohydrate on glycemia and insulinemia for ultimately for the development of medical foods for people with diabetes. And I thought, wow, research is really fun. You get to ask questions that have never been answered before. And so I pivoted again a little bit and decided that I wanted to go into research. So as a research dietitian, I took a job also at Ohio State as a clinical research coordinator for a few years. I was able to be involved in every aspect of development, recruitment, retention, data collection, nutrition education, data management, analysis for 2 different clinical studies, one related to nutrition for healthy nutrition for aging and one related to dietary oil supplementation and its effects on adiposity, body composition, and glycemia in people with type 2 diabetes. And so I really enjoyed that opportunity, and I also became more curious about the whys and the hows. So I, in the clinical research that I was doing, we were able to answer, does oil X, have physiological effect Y? And so we could answer those questions, but I was becoming more curious about the whys and the hows. So why does this dietary oil improve glycemia? Like, what is the molecular mechanism? What happens inside the body? And so that's where I started getting the PhD bug and decided to, despite being a first-gen student and really not even having the confidence that I would finish college, here I am trying to decide whether to pursue a PhD, and eventually made that decision. So I was also at Ohio State during that time in an interdisciplinary nutrition sciences program, and under the mentorship of Dr. Martha Belluri, and in collaboration with the Dennis Guttridge Lab, looked at cancer cachexia and some of the metabolic work that I had previously done really looked at the role of insulin resistance in the development and progression of cancer cachexia in the C26 cachexia model. And during that time, I was also able to collaborate on a project with another student in the lab. She found that there was decreased cardiac function in vivo, looking at echocardiography in mice. And so that really, I became very curious about the heart, and I was introduced to Dr. Joe Metzger at the University of Minnesota, and so ended up back home. Minneapolis is my home. So back to Minnesota for my postdoctoral training in his lab in the area of cardiac physiology and molecular cardiology. And there I did a number of projects related to gene therapeutic strategies for calcium mishandling in diastolic dysfunction. And so in my last year of my postdoc, I wanted to revisit the cachexia story. So I wanted to determine whether there was uh, heart organ intrinsic dysfunction and cardiac myocyte intrinsic dysfunction in this model of cachexia. And that's what I did sort of during the last year that I was in Minnesota. And that is really what I'm building here at Baylor. But before that time, so that's how I became, I don't think I can get through college to I'm doing a postdoctoral fellowship, and then now my journey continues from the North Star State to the Lone Star State. So during my postdoctoral fellowship, I met my life partner who was in Wisconsin, and we decided eventually we wanted to do our life together. So when he was offered a position at Baylor University, we made the trek literally down I-35 the entire way to Waco, Texas. And so that was a really big pivot in my road. But I spent a year going back to my clinical roots, doing clinical research management at a major medical center down here. And then after our first daughter was born, made the jump to the classroom, which I wasn't really entirely sure how I would like classroom teaching, but I thought it was a lot of fun, and it was a huge learning curve for me. But once I got into it, I really started to enjoy that. So during that time, our first daughter was born, our second daughter was born, and I was also working to publish a couple of papers from my postdoc that I hadn't quite gotten out the door when I had left Minneapolis. And so then I was really just so grateful for this opportunity that came along to apply for and be offered a position on tenure track here at Baylor. So I am currently an assistant professor of nutrition sciences at Baylor, and I started that role right after the pandemic started. So that's kind of a very momentous time in our world's history and my history as well. Here at Baylor, I have a 50-50 position. So I teach 50% of the time, and I do research 50% of the time. I teach 2 undergraduate and 2 graduate courses. I teach an introductory nutrition science class and then an advanced nutrition metabolism course for undergrads. And then I teach a graduate series, macronutrient and micronutrient metabolism. So when I'm not in the classroom, I do research. And so I'd taken quite a number of years off of active academic research, but found myself right after the pandemic started rebuilding. And it was an exciting time. Yeah, super exciting time. I was just so grateful for this opportunity. And so that was my journey from the North Star State to the Lone Star State. And we are still here in Texas. Funny enough, I love snow. I'm a huge fan of snow and cold weather. And I moved to Waco in August. So I really felt like I couldn't breathe for the first, 2 months that I lived here whenever I stepped outside. But many years later, I'm adjusted. So I don't enjoy August here, but I can tolerate it pretty well. And so I count that like a huge win for me. So that's my journey. That's how I got from Concordia College to my current position. And I never in my wildest imagination as a freshman at Concordia would have guessed this is where I would be. But I love it. I'm grateful for this journey, this windy road that I've been on. And so that's the journey. And now I'll share a little bit about my research areas to kind of transition here. My primary research focus, just because of my PhD and postdoc experience, I really kind of combined my training and expertise to a new niche area that really hadn't been explored in any great detail related to cancer, cachexia-associated cardiomyopathy. I do have a lot of other, not a lot, but a handful of other projects going on in the lab and other interests as well. As I have been told, never put all your eggs in one basket, and I am a firm believer of that. But I also think that it's not good to put, one egg in 40 different baskets. So you need to be wise, keep your eye on the goal, but also be willing to branch out a bit. And I think that particularly, it's taken me a long time, really, because of a lot of challenges that have, little dips in the road and things have caused me to take quite a bit of time to really get the heart research active and up and running here at Baylor. And so that's really given me the opportunity to branch out and to meet other people and collaborate with other people across campus. And so we have projects going on related to looking at different dietary interventions as a dietitian and changes in the microbiome, the intestinal microbiome. We have a couple of projects related to the difference between young and old animals with cachexia, so aging effects on muscle and adipose wasting, and then the effects of cachexia in the context of obesity. And we also have an underlying interest in also exploring sex differences as well. But my main area really is the cardiomyopathy. And again, it's taken me a while to really get that going, but we're doing it. So, if you have a delay, don't give up hope. Just keep moving forward and taking one step at a time. So we've got a lot of things in the pipeline, and we're getting ready to get some papers out this year. We're really excited about that. But so I don't really need to introduce cachexia to this group at all. I realize that. But to date, most research has really focused on skeletal muscle. We do know we are branching out and realizing that cachexia is very, systemic and affects nearly every tissue and organ in the body. And the heart is one of those organs. We do know from my past work at Ohio State that the heart does atrophy. And this data was published a long time ago, and it was in the C26 model. You can see here there's about a 20% decrease in the mass of the heart. So it atrophies very similarly to the skeletal muscle. And when we look at in vivo function via echocardiography, we see a significant blunting of fractional shortening. So there's an in vivo functional deficit. And so this has been found in multiple other published works in preclinical models, and also some clinical literature showing that patients with cachexia have diminished cardiac mass and cardiac function. And so therefore, knowing what we know about the heart, the heart delivers oxygen to our tissues, and if our heart isn't working well, that could actually have a really important and significant role in the fatigue and weakness that patients with cachexia experience, and could actually have a role in cachexia pathogenesis, as we do know that heart failure patients a lot of times do develop muscle wasting disorder as well. So like I said, this was at Minnesota, but when I went to Minnesota during the last so this happened at Ohio State, and then this was more in Minnesota during the last couple of years of my postdoc. I was curious about whether there was heart intrinsic functional deficits, or whether that the fractional shortening deficit was due to some systemic factors. So I'm not showing this data today, but I took the hearts out of the mice and looked at whole ex vivo cardiac function and found a greatly diminished pressure development and positive and negative pressure derivatives, which indicates both systolic and diastolic dysfunction. And then I went a step further and took the myocytes out of the heart. And so to do this, we perfuse the heart with a small with a physiological solution. You can see here it's oxygenated. It has everything the heart needs to survive, but we also put a collagenase enzyme in there, and that breaks apart the extracellular matrix. So we can see the heart cells kind of coming out of the heart. And so this is what a cardiac myocyte looks like. We can stimulate this cardiac myocyte with a small amount of electricity and look at the contraction and relaxation. And these are just some traces. This represents a full shortening or contraction of the myocyte, and then a re-lengthening or relaxation of the myocyte. And we can also load these myocytes with the calcium indicator dye, which fluoresces at a different wavelength when it's bound to calcium versus unbound to calcium. And so we can measure in conjunction with the contraction and relaxation, we can measure the calcium coming into the cell during the contraction phase, and then the calcium decay during the relaxation phase as well. And so we found very significant blunting of myocyte function, as this is just a representative trace over here on the left. You can see compared to myocytes from control animals, which have a very robust and strong contraction and fast relaxation in the black bar, myocytes from tumor-bearing animals in the dashed bar, the dashed line here, have a very blunted contraction, maximal contraction, in a very slow time to peak contraction and time from peak to relaxation. And you can see that data quantified over here. So we have a decrease in peak shortening and also a slow contraction and a slow relaxation. So these cells are really, I say that they're weak and they're slow. And so we wanted to see whether calcium had anything to do with that. And so we loaded these cells with a calcium indicator dye and found that the very similar type of trend. So again, just this is the calcium coming into the cell in the contraction phase, and then the calcium decay during the relaxation phase. And you can see from the myocytes from tumor-bearing animals that the peak calcium is decreased and the calcium decay is slowed. And you can see that quantified over here with a decreased peak calcium, and then a prolonged time to peak and prolonged peak to 50% decay. So we've done some preliminary work with looking at like beta adrenergic sensitivity. We've looked at some calcium handling proteins and not found really substantial differences. And we moved into more of the metabolic effects. We know from published literature that mitochondria dysfunction has a role in skeletal muscle atrophy. And so we hypothesize that mitochondrial dysfunction may have a role in cardiac atrophy and dysfunction as well. So the heart accounts for a huge percentage of our basal energy expenditure, about 10%. Despite making up only about 5% of the total weight of an adult human. Mitochondria are very important. They make about 90% of the ATP utilized by the heart, and the heart has a constant need for ATP. That muscle never rests. And so mitochondria make up about a third of the total cardiac myocyte volume. And so we collaborated with the Michael Wiggs lab and looked at mitochondrial respiration in mouse hearts with tumors compared to controls. And we found that there's a trend towards a decrease in oxygen consumption in hearts from tumor-bearing animals, and a significant increase in reactive oxygen species, which is a marker of oxidative stress in heart tissue. And of note, this is in male animals. Most of our previous work had been in males only, but our female mice are exhibiting a different phenotype, and that is really what has gotten us interested in sex differences. And so I'm not showing any of that data today, but just we are kind of diving into some of those potential differences and why we're seeing those differences. Additionally, I'm just showing you a smattering of the gene expression data that we found. We found decrease or alteration in mitochondrial dynamics in fission and fusion, and autophagy and mitochondrial biogenesis markers. So we are moving forward with trying to understand a little bit more what's going on metabolically with these hearts, and if that is contributing to the cardiac dysfunction. And so the conclusions: we've distinguished isolated cardiac myocyte functional deficits from systemic factors, and calcium cycling deficits underlie the contractility and relaxation dysfunction, and mitochondrial deficits are potentially contributing to calcium mishandling. Most of the energy utilization by the heart is the myofilament crossbridge cycling and the calcium cycling as well. So that is our hypothesis moving forward. And next steps really trying to in multiple different ways correct mitochondrial function in these hearts and see whether we can connect more specifically mitochondrial function to mechanical functional deficits by measuring the myocyte function, contractile function, and then also looking at sex differences as a way to determine, mechanism related to what is protecting females to a greater extent than males. And so that's really my main research agenda. There's, it's still, I would say, in the fairly beginning stages, but we're moving forward with it, even though it's taken a while. And so that's really exciting. I guess, put one foot in front of the other and keep doing the next thing. So that's where we're at, and we're looking forward to some papers getting out here in the next year or so. So from here, I guess I'd like to just end probably talking a little bit about some of the lessons I've learned through my journey. So I wrote down, I would just kind of brainstorm things that, that I learned throughout these years. And I thought, I would have a few, but I actually have 9. So I'm really sorry. I wish that I could make this really succinct, but I wasn't really sure how to make it less, because I think they're all very important things that I've learned. But the first thing would be just to hold your plans loosely. I know it's really cliche to say this, but life is a journey, not a destination. I think it's important to have goals. It is important to work towards those goals, but to hold them with open hands and not become so laser focused on the ultimate goal that you're missing out on other opportunities. I would encourage you to pay attention to what excites you and interests you, even if it surprises you a bit. Like for me, as an undergraduate, really enjoying chemistry, that surprised me quite a bit. I've had quite a windy road, but I've had opportunities to really step outside my comfort zone and try something new, and I'm grateful for those opportunities. I'd say the next thing is just accept that there's a lot of things you can't control, and that's scary for me who likes to control everything, but you don't have control over who reviews your manuscripts or your grants. You don't have control over the economy, the political climate. You don't have control over leadership and direction changes at your company or your medical facility or your university or wherever you're working. The job market, how how many people are being hired in a particular year, health issues that you might have or your family members might have, or even like really little simple things like what mood your kids are in when you pick them up from school. You think you're going to get a lot of work done in the evening, and then that gets quickly hijacked. So I think if that scares you, that's pretty normal. It can scare me too, but I think really focusing on what things you can control, and letting go of the rest. Just being consistent and working hard and doing the next thing and trusting the outcome. I think if you try to hold on to everything, it just becomes very frustrating and it's not worth the effort. I think for me, one big thing is that the closer that my expectations are to my reality, the happier that I am. So I tend to be a perfectionist and I tend to have high expectations for myself. And I always have this idea that I should be able to do this and I should be able to accomplish that. And when what you think you should be able to do is you're always falling short of that, to kind of revisit what your expectations are and are they realistic given the reality of your life as a whole, like all of the other things in your life that are going on, and what is actually realistic for you to expect of yourself. So I think sometimes it's easier to change our expectations than our reality. But clearly, if you're very unhappy with your reality, that can be changed too, but that can oftentimes take more time and more effort. I would say another thing related to that would be that just know that you can't do everything. Some people will tell you you can have it all, and I'm a firm believer that you can't. I think we all get 24 hours in the day and we make choices with how to spend our time and what's most important to us. And we can't be the best at this and the best at that and the best at this and the best at that. We need to make hard choices sometimes about what is realistic, and what is feasible based on our current life, and also what is most important to us, our values. So I think the more that I've been able to accept that, the more I'm at peace with things. I think success is a combination. It's obviously a lot of hard work. It's a lot of intelligence, and it's a little bit of luck. So I think thinking that it's all just being really smart and working really hard can maybe make you think too highly of yourself or too lowly of yourself, depending on how lucky or unlucky you've been. So I think that when opportunities arise, to have the intelligence to recognize that those are opportunities, and then the hard work to follow through with those opportunities is probably the best way to go. Be confident and humble. I tend to not be overly confident. So, you're smart, you work hard, you've accomplished a lot, you have something to offer this world. And so believe that, like, truly to the bottom of your soul. But also know that no one knows everything. No one knows all the answers. And so I think for me, it's been always very helpful to listen and consider the ideas and opinions and perspectives of others outside of my field, and with different walks of life. I think a lot of wisdom for me has been gained by just simply listening to others. When you run into roadblocks, pivot, but don't take your eye off where you ultimately want to be. I ultimately want to do to understand cachexia associated cardiomyopathy, but I've had a lot of twists and turns in my road and different roadblocks. And sometimes when you're, in front of, a brick wall, maybe don't just keep trying to hit it and, like break through it, but just walk around it or, like think of a different direction you can go or how you can climb over it. And pivot and do things that can keep you moving forward towards where you ultimately want to be. On the bad days, and there will be many, tell yourself what you have done well, because there's always something that you have done well. Even when it's like the whole day is like a dumpster fire, there's something that you did well. And so cling on to that and move forward with that. Prioritize, and then stick to your priorities. I actually list out my core values. What are the most important things of my life and how I can live those out tangibly in my day-to-day life. And I revisit them often. And so I can't do everything. I mean, I'll take like the state of my house, for example. I would love to redecorate my house and to make it pretty, but I have two young kids at home, dual career couple. We don't, it's not my core value. Redecorating my house is not my core value. It is a want, but it is not a need. And so, when I look at that and I get frustrated, I think, but how am I spending my time and what are my core values? And have I gotten off track? And if I've gotten off track, how can I pivot back? And then just being at peace with knowing that, when you're not getting everything done, and you will always have more things to do than you have time for, but that you have put your priorities first. And then the last thing I would say is pay it forward. None of us are self-made. I have had many mentors and teachers and people that have spoken into my life in different times and ways and places. There's been people that have, taught you things, provided you with tools or information or other tangible information. So to be grateful and live in gratitude, I think would be to pay it forward and to help those, help others around you. Your legacy is not simply what you accomplish, but also what you help others to accomplish as well. So those are some of the lessons learned. I guess that's kind of my talk. I talked to you about what I have, my journey, where I got to where I am, kind of what my research is and what I'm currently doing and why, and some of the lessons I've learned along that windy road. So I'm happy to take questions. Let's see from anyone. I will shut this down and let's see, I'm not sure how I get this bigger. Okay. So Q&A, do I go to the Q&A or do I go to
Yeah,
the
I
chat?
can. If you can't see them, I can read them out to you. You've got a lot of questions coming
In
in.
the Q&A, I see
Yeah,
some.
they're in the Q&A.
Okay.
Do you want me to read
Yeah,
them
I
out or
see
can
them.
you see them?
I
Okay.
can see them.
Okay, great.
Uh, so there's a question on ages of male and female mice. We generally, for our young animals, we're using around 10, maybe to 12 weeks old generally. But our older animals, 18 to 21 months is usually when we study them. Um, and we use both males and females for most of our studies now. How do I want to correct mitochondrial function? That's a really good question. We are looking at a couple of different approaches, both a genetic approach and a couple of different uh, chemical or pharmaceutical approaches. So we're currently seeking funding for that and doing a little bit of pilot work, but we're moving towards both pharmaceutical and genetic approaches for that. As far as, let's see, I was wondering if there might be any differences between cardiac chambers, particularly left versus right ventricle. That's a really good question. And I really don't know because we're studying the heart as a whole right now. But as far as like if there's difference in gene expression or protein expression or mitochondrial function in the left versus the right, is a really interesting question and something that we could definitely move into when we get more funding and time. Great talk. I'm also an RD. Yay. Let's see. Are you doing any heart nutrition related research? Not heart specific right now. We are looking at some multimodal dietary intervention on cachexia outcomes in general, and we obviously will be looking at the heart, but that's not our main hypothesis. So our dietary intervention is focused on mitigating inflammation and also the imbalance between muscle anabolism and catabolism. So it's not a cardiac specific approach. Is cardiac mitochondrial dysfunction correlated to muscle mitochondrial dysfunction? I'm not really sure exactly what that question means, but so there was a paper published that found mitochondrial dysfunction in skeletal muscle was an early event. So it actually was happening prior to the onset of really significant muscle wasting. We have not found really significant early mitochondrial dysfunction. In the heart, it seems to be happening a little bit later in the pathogenesis. For that, let's see. I'm wondering if I'm not fully answering questions, please feel free to speak up and ask additional questions. I don't want to write people off. I feel like I'm reading the question and doing the best I can with what I have. So
yeah, I think you're doing great. But if anybody wants to ask questions live, you can raise your hand and I can unmute you.
Okay, biomarkers for cardiac cachexia. I could measure retrospectively in plasma samples in my patient cohort. Gastrointestinal cancer patients with and without cachexia. I mean, your acute cardiac damage, yeah, your BNP, your troponin would be your best, but I'm not sure. We haven't measured those in our mice. We don't suspect that, those are, I think BNP might be one. That was shown in a clinical, in patient samples. I don't remember the year, maybe 2014 or so, there was a paper that found increased BNP in cancer patients. But that's a little bit tricky because I think some of it's like, like an MI or something would be like an acute damage, and or pressure overload or, ischemia reperfusion or something would be like major damage that would cause like a huge upregulation of some of these markers. I don't know if it'd be more nuanced in cardiac cachexia.
I think that Brittany raised her
Brittany?
hand. If maybe
Okay.
she wants to jump in and ask a question.
Yes, I think you can hear me. So my question is, going from an adjunct to an assistant professor, what was the biggest hurdle? Or is that process, I don't know. I think especially with maybe the climate now that there might be a lot more adjunct positions available than assistant. So I think there might be more people in the future going through these same steps.
Yeah, so for me, I didn't know. when I moved to Texas, I wasn't sure. I knew I wanted to be involved in research if I could. And so I did go back into clinical, so like a medical facility. It was not an academic position, but it was helping physicians with clinical research projects they were interested in looking at with their patients. Just a lot more like retrospective chart review and prospective like survey types of things. And that I was commuting a lot. You know, when my daughter was born, it was not feasible. And so I stepped into the classroom for the first time, and it was a huge learning curve for me. But I would recommend it because if y'all like, I was a RA as a PhD student and a, you know, postdoc. I guest lectured just a handful of times and did some tutoring, but really no like teacher of record classroom experience. It's hard when you start. And so I just felt like it was so invaluable for me to get that experience before I started on tenure track, because then once I dove into tenure track, even though I'd taken several years off and I had to totally rebuild my research agenda from ground up, I at least had like the context for how to teach classes. Granted, I had 3 new course preps my first year on tenure track, which was crazy, but really if I had never been in the classroom before, I don't know if I would have survived. So it was really helpful for me to get that experience. For me, it was just a lot of networking. I think here at Baylor, I was really fortunate to get to know the faculty in the department when I was teaching these classes, and they knew where I had been. They knew my history. They knew that I was interested in moving into a research position. And so when there were a couple of retirements, then I obviously put together my package, and I think it was, you know, they knew who I was and they knew, you know, I had potential. But I would say if you're, you know, if you've done only research, and you want to do academia, if there's any way you can get teaching experience, it would be really helpful. Is that helpful? Does that answer your question, or is it?
Yes, it does. Thank you.
Okay. Dina, I think, had a question.
Yeah, I did have a question, but I want to highlight a question from somebody that's kind of in the same vein. Do you have a solution if we can't continue the journey in science? These days, it's so difficult to get a position. I think this person is asking for any recommendations you might have.
So, I mean, there's obviously, you know, everyone that like they go to grad school thinking they're going to get a postdoc and then a tenure track position. And they're really hard. They're really few and far between, right? And the funding climate is really difficult right now to get funding. And if you don't get funding, you don't get tenure. And so, I mean, I'm in that position now. I'm an assistant professor on tenure track. So I hear you. And I think just trying to explore like what gets you excited. I've considered, because again, my husband is an academic, and so we have this 2-body problem, and we determined we're going to do this life together. And other people might be different. Other people do, you know, live in 2 different places and all of that. And that's, you know, again, core values. You need to go back to your core values and what is most important for you at a given time. But I have, you know, like I said, I did, you know, clinical research management. I've done, I've been interested in like medical writing or scientific writing, teaching. I mean, I think there is always a demand for teaching. It's unfortunately, I feel like in this climate, it gets a little bit difficult with like, I don't know how I don't know how long this climate is going to last, and I don't know how it's going to affect higher education long term. I would say that we can't jump to any conclusions yet. There's still a lot of college students that need an education. So there's a lot of people that need to be teachers, but whether or not universities react in ways that are like, doing less like permanent positions and more adjunct. And I think online education is getting to be bigger and bigger. So, in that situation, you might be able to teach a number of different classes from anywhere in the country. But then also looking at industry and what different industry, what your expertise is, and different industry options for research, or any phase of the research process from like, early phase all the way through implementation. And yeah, I don't know. I would say don't give up on your dreams, but sometimes, like I said, I pivoted so many times, and I've taken a lot of kind of side roads, and I still might take some side roads. Who knows? Like I said, I hold my plans loosely, and I control what I can control what I can control, and I have to give up the rest because otherwise I start freaking out. So yeah.
I think there's one
Um,
more scientific question from Daria. Wondering if you have looked at cardiac mitochondrial calcium retention capacity.
That is something I would like to do, but no, I haven't done it. Because that has to do with mitochondrial health clearly, and calcium overload. And yes, so the answer is no, but yes, I would like to. Thank you for your question. It's good to hear other
Great.
assistants professors feel like that. I hope students and postdocs can be a step ahead knowing this. Thanks for your topic. And okay, anything else?
Yeah,
It
are
was
there
very
any
inspiring,
other?
Michelle. Thank you for taking us through your journey. It was very inspiring. And
Yeah, and if anyone has any questions or wants to chat offline, I'm happy to. I mean, like I said, I'm not a super formal person. I'm happy to share more details of my stories and my life. It's crazy trying to raise a family and have a career and have a, husband in academia, and it is crazy. And so some days you feel like you're not like even keeping your head above the water, but sometimes like I'm just like I'm still alive. I'm doing well, so anyway, but yeah.
Great, sounds good. Thank you so much for your talk today. We
Yeah,
really
thank you for
appreciated
having
hearing
me.
from you, and it was great. And thanks for everyone who tuned in and stayed for all the questions at the end. We'll hope to see everyone again sometime soon.
yeah, thank you,
All
everyone.
right.
Thank you so much, Michelle.
Thank you very much. Have a great
Bye.
afternoon.